coming for yet another segment of Homeschool. I am your teacher, Mr. Lwanyaga Solomon, and I will proceed with what he did yesterday. So if you are new to this segment, this is a segment supported by Grace and Life Church Ministries, Code High School, and our beloved pastors, Pastor Jackson Senyonga and Pastor Evelyn Senyonga. We welcome you to visit our website, www.codehighschool.sc.ug. Go there, sign up, get an account, and access all the information that we are talking about. And also endeavor to read as a biology student, endeavor to read as much as possible, endeavor to engage yourself with acquiring as much knowledge as you can. Yesterday, we started on a topic called coordination. And we said coordination is the way different parts of the same object work together in harmony to have a smooth action. For example, in a factory, we have very many parts, very many individuals doing different things, but these individuals need to work together so that they are able to produce something that is uniform, something that is good, something that is unique. Likewise, our bodies and plants also need to be coordinated. Different parts of the plant need to be coordinated. The cells need to coordinate to form tissues. The tissues coordinate together as organs, and the organs make systems, and the systems make an organism. Deep down, we need to have a mechanism of controlling these different effects such that we can have a fruitful result, and this is what we are going to look at in this segment. So yesterday we looked at two types of coordination. If you take me to slide number 2, we looked at two types of responses, and we said plants respond by moving. Remember, we know that plants are unable of locomotion. They are unable to locomote. Therefore, we know that plants respond by moving. And we have two types of responses. We have nastic responses and tropic responses. Under nastic responses, we have very many responses that we talked about, but we emphasized thigmonasty, which is response to touch, and photonasty, which is response to light. We gave an example under thigmonasty of the Mimosa pudica, the touch-me-not plant, and we looked at the mechanism of how its leaflets droop when it is touched. We gave an example of the Venus flytrap. We gave an example of the sunflowers. We talked about the petals of flowers, how they close at night and they open during day. I also hinted on seismonasty, the response to pressure, and I said this also is an example of a nastic response. We also said nastic responses are not growth responses. In other words, a plant does not need to grow to respond to a nastic response. Tropic responses, we also talked about tropic responses. We gave quite many examples, but we delved into the matter of phototropisms. We talked about how the different scientists responsible for the development of the science of phototropism, who discovered, who experimented. We talked about Charles Darwin and his son Francis. We talked about Boyce and Jensen. We talked about F.W. Wendt. We talked about what they contributed to the development of this science. Now, today, these are the lesson outcomes that we expect today. So if you go to the next slide, I want us to look at the lesson outcomes. We expect to study the following things today. We are going to look at photoperiodic responses. We are going to look at photoperiodism in particular. We are going to study the different scenarios of photoperiodism. We are going to look at geotropisms, hydrotropisms, and chemotropisms. So very quickly, go with me to what photoperiodism is. So photoperiodism, yes, we are saying these are physiological changes that occur in plants in response to relative length and day and night. Plants respond, I said yesterday that plants respond to the time of the day by responding to light. So we are looking at photoperiodism. So we are saying these are physiological changes. Let me say these are changes in plants These are changes in plants that occur due to a change in day length. In other words, photoperiodic changes photoperiodic changes occur when the length of the day changes. We know that we don't entirely have the same amount of time in all throughout the year. We have months in the year where we have longer days than nights, and we have months in the year where we have longer nights than days. And plants have also adapted to be able to respond to these different physiological changes. So their physiological changes are sometimes coined to the period of day. For example, we know that flowering does not happen throughout the year. The reason flowering happens at particular times of the year is exactly this, that photoperiodism, that plants are going to track light and the length of how much light they have received to be able to produce light. So under this, we look at the scientists Gunnar and Allard in 1920, just a few years after Boysen and Jensen did their experiments. Gunnar and Allard carried out experiments on photoperiodism. And they discovered three types of plants, three classification of plants. We have short-day plants. We have short-day plants, and we are going to talk about more of all of this. We have short-day plants. We have long-day plants. And we have day-neutral plants. Day-neutral plants. And short-day plants are plants that require a short period of day length, a short period of daylight, and a longer period of night. So some people actually call short-day plants long-night plants, while some long-day plants require a longer period of daylight so that they may be able to flower. This is why some people call them short-night plants. Day-neutral plants are plants that can survive in both conditions. In periods where they have too much light and periods where they have short daylight, they can be able to produce flowers. These are the three categories that they discovered. So very quickly, if you go into the next slide, I want us to delve into the matter of short-day plants. Yes, so we are saying short-day plants are plants that require relatively short daylight period, sometimes a minimum of 8 to 10 hours. They require a minimum of 8 to 10 hours. If you see in that illustration, we see that in short-day plants, 8 hours of light are sufficient for these plants to flower. 8 hours of light are sufficient for these plants to flower. However, experiments were done onto the short-day plants, and we are going to talk about that more. Some examples we have there: we have rice, we have coffee, we have soybean, tobacco, and chrysanthemum. Yeah, most of the common examples we know. We know rice, you've seen rice, you know coffee, we know soybeans, tobacco. These plants require a period of at least 16 hours of night, at least 14 to 16 hours of the night. If they are not provided with this period of night time, they may not be able to flower. And because of this, they are called long night plants, because it was realized that an interference in the daytime may not have as much effect as interfering with nighttime. Yes, with you see that illustration right there. We see that short-day plants require 8 hours of light and 16 hours of darkness so that they are able to flower. When you look at the next bar graph, the night time was interfered with a brief period of light. Yes? And this light is not just any light. I talked about this yesterday, that plants don't just absorb any light. They absorb particularly in the blue and the red region. They don't absorb green and yellow light. They absorb mostly in the blue and the red region. So when they interfered this period, this 16-hour period of light with halfway in the middle, when they interfered it with light, plants did not flower. However, when they interfered the daytime with a period of darkness, short-day plants were able to flower. This tells us that these plants require more of the darkness. It is more dependent on the length of the dark time than the length of the daytime. And we are going to delve into why this is so. Short day plants, yes, require a shorter day period, but this is to tell us that they cannot work without a long period of darkness. So when you look at that illustration there, we have the night time. When the night time was interfered halfway, when it was interfered with a brief period of light, the plants were unable to flower. Yes. However, when the daytime was interfered with a period of darkness, these plants were still able to flower. Okay, so let's look at the long day plants. What is the case for the long day plants? If you go to the next slide. So the short day plants, the long day plants, are the exact opposite of short day plants. While the short day plants require a longer period of night, long day plants require a shorter period of night. And while the short day plants require a shorter period of day, long day plants require a shorter period of day. So we are saying these are plants which require a longer day period, around 14 to 16 hours, for them to be able to flower. They need a lot of light for them to be able to flower. But I want to emphasize that in photoperiodism, it is more dependent on the period of the night than it is the period of day. And we are going to assess this. We are going to find out more of these. Examples, we have cabbage, we have wheat, we have sugar beet, spinach. We know some of these plants. We know wheat, we know cabbage, we know sugar beets. So we can also call long day plants short night plants. Now, unlike short day plants where the long night period is critical, in day plants the daytime, the light period is critical. Interfering with the day period may cause a brief interfering with the day period may cause a failure of flowering. Yes, because remember, these plants require a shorter period of the night. So the longer the darkness, the more they are going to fail to flower. The longer the darkness, the more they will fail to flower. So we are saying that a brief exposure of red light in the dark period or the prolongation of light stimulates flowering. In other words, if you expose these plants to increased periods of light, and this light should be particularly red light, if you expose them to increased periods of light, they are going to be able to flower. And this knowledge is used a lot in our gardening today. Florists use a lot of this knowledge to be able to produce flowers throughout the year. They manipulate what period of time they provide for their plants. So we are saying short day plants require shorter periods of day and require longer periods of night. Long day plants require longer periods of the day and shorter periods of the night. And however, the period that is critical is the night period. Interfering with the night period for the long day plants causes flowering. It stimulates flowering. If you have darkness, then you put light in between there for the long day plant, you are going to have flowering. However, for the short day plants, if you interfere with the night, you're going to inhibit flowering. Yes? If you subject long day plants, if you interfere with that day period, you are going to, and you will not be able to produce light. So you will not be able to produce flowers. Because for them, the dead time is critical. The dead time is critical. We are going to assess why that is possible. Very quickly, if we look at the denuto plants, yes. Like I said, these are flowers. These are plants that flower in all photoperiod ranges. They can flower when they are provided with 5 hours of light, and they can flower when they are provided with 24 hours of continuous exposure to light. They flower all the time. Good examples, we have tomatoes, we have cotton, we have sunflower, we have cucumber, we have peas, different varieties of tobacco. Yes, all these are plants that do not exactly depend on how much light they are provided with. They rather depend on, they do not depend on the photoperiodic range. They can flower when they're provided with little light or when they're provided with too much light. Yeah, so I want us to try to think about this. In recent studies, we have discovered new types of plants. We have long short day plants and short long day plants. Yes, we have discovered short long day plants and long short day plants. And I want you to go out there and think about them. and think about some of these things. We have discovered quite a number of things. We have discovered long, short day plants and short, long day plants. Go out and find out what are long, short day plants. Is it a confusion? Yes, those are plants actually that are out there. So write them down and go and find out. Then the trial question I want you to write for me and put for me on the website is identify the differences between short day plants and long day plants. This should be quite simple for A-level students. Now, I want us to look at, we have seen the differences. We have seen that short day plants flower when they are provided with longer periods of night, and long day plants flower when they are provided with shorter periods of night. What I want us to assess next is what is the underlying factor for these plants to behave like this? Why do they respond like this in these periods of time? The material we thank for, the substance we thank for that is the phytochrome. When we want to assess why these plants respond like this, we look at a material known as the phytochrome. Now, I talked about the phytochrome yesterday, just that I didn't mention this word. I said the substance responsible for receiving light in plants is a substance that is joined to a protein, a chromophore joined to a protein. I talked about this yesterday. I talked about a chromophore joined to a protein, forming a chromoprotein. Forming a chromoprotein. Now, this chromoprotein is what we call the phytochrome. If you take me to the next slide right there, the next slide. So the chromoprotein is responsible for receiving light. And we are saying it is present in the plasma membrane of cells. Yes, it has two components, the chromophore and the protein, forming what I said yesterday, known as the chromoprotein. Now, the pigment, this phytochrome, exists in two forms. It exists as phytochrome red. It exists as phytochrome red, commonly known as PR. Phytochrome red absorbs light around 630 nanometers, 660 nanometers to 665 nanometers. It absorbs light within that wavelength, and this wavelength is red. So it absorbs red light. It absorbs red light. However, also, phytochrome, so we are saying this one absorbs light in this wavelength, and it also absorbs red light. This phytochrome also exists as the pigment phytochrome far red. Follow me, phytochrome far red. Phytochrome far red commonly distinguished as PFR. PFR absorbs light in the wavelength 730 and 735. (730 and 735). And phytochrome far red absorbs far red light. Phytochrome far red absorbs far red light. It is very, very important. Now, you should also know that these two phytochromes are interconvertible. They are interconvertible. What does that mean? It means that if we have phytochrome far red here, phytochrome far red can become phytochrome far red. So phytochrome red can become phytochrome far red. And likewise, phytochrome far red can become phytochrome red. When does this happen? When phytochrome red absorbs red light, yes, during day. Red light is very common during day. Yes, it's very common during day. So when phytochrome red absorbs light, it is converted into phytochrome far red rapidly. It is rapidly converted into phytochrome far red. When phytochrome far red absorbs far red light, in other words, this is red light of long wavelengths. Yes, it absorbs far red light. It is also rapidly converted into phytochrome red. However, phytochrome far red retains the ability of becoming phytochrome red gradually in the dark. That means even in the absence of far red light, phytochrome far red can be converted. So this is in the dark. It's in the dark. So phytochrome red, when it absorbs light, it is converted into phytochrome far red. And when phytochrome far red absorbs far red light, it is converted into phytochrome red. However, in the dark, phytochrome far red can gradually convert back to phytochrome red. Very important. So when you take me back to that slide right there, yes, we are saying phytochrome red, when it absorbs red light, it is converted to phytochrome far red. And when phytochrome far red absorbs far red light, it is converted back to phytochrome red. However, in the dark, phytochrome far red can be converted gradually back to phytochrome red. That is why we have that sign there being carved, indicating that it takes quite a long time. These other two happen rapidly. Now, let us look at the effect of phytochrome on short-day plants. Yes? What is the effect of phytochrome on short-day plants? Now that we have seen this, let us assess how it affects short-day plants. Why is it that short-day plants require a short-day period? The first thing we need to note is phytochrome far red is the active component of the phytochrome system. It is the one that is responsible for the reactions we are talking about. Phytochrome red is inactive. Yes? Now, remember we have said that phytochrome red absorbs red light during day. Yes? Yes, please put me back on the screen. We are saying phytochrome red absorbs red light. Let me make this a bit bigger. So this is phytochrome red, phytochrome far red, and this is red, this is far red light, and in the dark this one becomes this. So this is dark conversion. Now, I have said that phytochrome far red, yes, phytochrome far red is the active form. Therefore, the presence of phytochrome far red is the one that initiates or inhibits flowering in plants. In short-day plants, large concentrations of phytochrome far red, this is quite important, large concentrations of phytochrome far red inhibit flowering. Large concentrations of phytochrome far red inhibit flowering. So, if after we understand that, let us look at this. This one, during day we have a lot of red light. That tells us that during day we have a lot of phytochrome red being converted into phytochrome far red, implying that during the day we have a large concentration of phytochrome far red, because a lot of phytochrome red is being converted into phytochrome far red, telling us that because phytochrome far red accumulates a lot during day, and remember it inhibits flowering in short-day plants. Short-day plants need to decrease the amount of daytime so that they decrease the concentration of phytochrome far red. That's the first thing. short day plants require a short day period to decrease on the amount of phytochrome red converted into phytochrome far red. Now, I also mentioned that short day plants require a long period of the night so that they are able to flower. This is why. During the night, phytochrome far red converts back into phytochrome red, implying that during the night the concentration of phytochrome far red decreases. That says the longer the period of the night, the lesser the concentration of phytochrome far red we are going to have. So short day plants require a short period of the day so that a little phytochrome far red is formed, and a long period of the night so that we have a lot of the formed phytochrome far red converting back into phytochrome far red. So if you interfere with this night, so yes, we had a short day. Yes, we had a short day right there. But in this period of the night, which is long, we expect that phytochrome far red is being converted, phytochrome far red is being converted back to phytochrome red. If you interfere with the period of the night and you provide red light again, the phytochrome red produced is going to be rapidly converted back to phytochrome far red. It is like washing and then you hang in the soil. You have converted, in this period you have been converting phytochrome far red to phytochrome red. When you provide light, all the phytochrome red produced is going to be again converted to phytochrome far red. So it is going to be as good as wasting time. So you providing phytochrome far red here, phytochrome red, providing red light here interferes with the conversion of phytochrome far red back to phytochrome red. And this is why interfering with a long night for a short day plant inhibits flowering. I also should inform you that if you provided far red light here, if you provided far red light after providing red light, far red light would counteract the effects of red light. Why? Because far red light converts phytochrome red rapidly back to phytochrome red. So providing far red light can counteract the effect of red light during the night. Take me back to my slide, please. So we are saying during daytime, the phytochrome far red form of the pigment is accumulated in plants. Phytochrome far red is inhibitory to flowering in short day plants. However, it stimulates flowering in long day plants. So the critical dark period in short day plants, phytochrome far red, the phytochrome far red converts back to phytochrome red, resulting into flowering because the concentration of phytochrome far red has reduced. Yes? A brief exposure with red light converts the phytochrome red back into phytochrome far red, inhibiting flowering. Yes, I have said that if you expose red light in this critical night period, you are going to be unable to produce flowers. However, exposing the plant to far red light cancels this effect of the red light because phytochrome far red will again be converted to phytochrome red. Okay. What is the effect on long day plants? The effect on long day plants, if we go to the next slide, the opposite is the opposite is true. For long day plants, we require a bigger concentration of phytochrome far red. When you look at it there, long day plants require a bigger concentration of phytochrome far red than they require a concentration than they do the concentration of phytochrome red. This tells us that for long day plants, they need as much as phytochrome red they can have converted to phytochrome far red to be able to flower. So we are saying they are prolonging the critical light period or the interruption of the dark period by red light in the long day plants stimulates flowering. Why? Because it results in the further accumulation of phytochrome far red. Long day plants depend on the concentration, on a large concentration of phytochrome far red to be able to flower. That is why the more red light they have, the more flowering will happen, because there is going to be a larger accumulation of phytochrome far red. Exposing the plant to far red light will cancel the stimulatory effect of red light because phytochrome far red will be converted to phytochrome red. So while exposing the plant to far red light at night converted phytochrome far red to phytochrome red and stimulated flowering in plants, in short day plants. In long day plants, if you expose the plant to far red light at night, the phytochrome far red will be converted back to phytochrome red and therefore will inhibit flowering. Okay. If we go to the next slide, I hope we have understood that. It's very important to understand how these two phytochrome systems work. Phytochrome red is inactive, while phytochrome far red is active. Some of those are the phytochrome mediated responses. Yes, some of those, most of all of those, depend on photoperiod to happen. How much light? Seed germination, if you didn't know, also depends on how much light we have available for the plant to be able to germinate. We have bud dormancy, leaf abscission, flower induction, protein synthesis in plants, oxygen catabolism, stomatodifferentiation, all of this. Now, I want to remind you that this work is there. You don't have to jot down a lot of things at the same time. You can simply go back after we are done with this segment, go and access some of this information on the website and make sure you access it. Make sure you access it. Okay, so we have realized how photoperiodism works. Photoperiodism works depending on the phytochrome system. It depends on the phytochrome system. We need phytochrome far red. The concentration of phytochrome far red determines if a plant will flower or not, if a seed will germinate or it won't germinate. Phytochromes are found in almost all the cells of the plants, because most of the cells of the plants need to respond to day period. Okay. So continuing with the next tropism, I want us to look at gravitropism. Commonly known as geotropism. Commonly known as geotropism. Geotropism. Geotropism, we are saying geotropism is response to gravity, and we are saying right there that shoots respond to gravity by growing upwards, and roots respond to gravity by growing downwards. The Darwins also are responsible for this science. They did experiments on geotropism, yes, and they realized, I like us to appreciate what these people did. They realized that removing the cap from a developing root stopped response to gravity. It stopped response to gravity. And the hypothesis we have for this is the statolith hypothesis. When you look at that picture we have right there, we have a cell having white substances. These are called amyloplasts. Amyloplasts are cell organelles that contain starch. Yes? Please show it, yes, thank you. Amyloplasts are cell organelles that contain starch granules. Now, because starch is denser than water, these cells are pulled to the bottom of the root cap by the force of gravity. In other words, if this is the root we have here, and this is the root cap, amyloplast will be pulled down here, like this. Amyloplast will be pulled down. If this same root is placed like this, if it is placed laterally, amyloplasts are going to be pulled down here. Yes? Now, according to the statolith hypothesis, we are saying the position of the amyloplasts activates sensory proteins. Yes? So when they come down here, when they concentrate down here, they activate sensory proteins that then result into the distribution of oxygens. That is the statolith hypothesis. The statolith hypothesis depends on special cells found in roots that contain amyloplasts. Amyloplasts have amyloplasts contain starch which is denser than water. And this starch is pulled to respond to the pull of gravity. They are going to go under they are going to be pulled down depending on where gravity is. If I place this one this side like this, these ones would be converted to come this side. Now, on every side they go to, they activate sensory receptors on that side. So ideally, what should happen? Take me to the next slide, please. What should happen? When you look at that picture right there, we have the amyloplasts concentrating down. What should happen naturally is that the amyloplasts should be down, and therefore oxygens will be distributed. Normal distribution of oxygens is going to result vertical root prior disturbance. So if you place if you place the root laterally, the distribution of the amyloplast is going to change. And because it is going to change, even how these oxygens are going to be distributed. Remember that distribution of oxygens depends on which sensory receptors have been activated. If the sensory receptors down are the ones which are activated, more oxygens are going to be distributed down than the oxygens that are going to be distributed up. What does that mean? That if it is this side where we have the amyloplasts, let me make it a bit bigger. If we have the amyloplasts distributed down here, because gravity is this side, we are going to have oxygens distributed more to this side. Now, the effect of oxygens in roots, we all know, is opposite to the effect of oxygens in shoots. While in shoots oxygens cause rapid cell elongation, oxygens in the roots delay elongation. So when oxygens concentrate here, they are going to cause delayed growth on the downer side of the root. They are going to cause delayed elongation on the downer side of the root. And that means the upper side is going the cells on the upper side will elongate much faster than the cells on the lower side. So as a result, we are going to have the upper part curving downwards. Because this side is not growing, we are going to have a resultant of something that looks somewhat like that. Why? Because this side that has a larger concentration of oxygens has delayed growth, and the side up is therefore going to grow faster. It's going to elongate faster than the side below. So if you take me back to that slide, yes, we are saying oxygens are actively redistributed. Oxygens are actively redistributed by the gravity sensing cells. The gravity sensing cells determine where oxygens are going to be distributed most. The side that has the largest pull of gravity is where oxygens will be distributed most. So we are saying gravity sensing cells, where the gravity is most, is where oxygens will be distributed most. Yes? And we are saying, as a result, asymmetric oxygen distribution, oxygens inhibit growth on the lower side, and therefore the upper side is going to grow faster, resulting into bending. It's very, very important. Take me back to the previous slide, please. When we look at the previous slide, I have talked about the Statholis hypothesis. What do you need to know about geotropism? Geotropism is response to gravity. And the major hypothesis we look at under geotropism is the Statholis hypothesis. The Statholis hypothesis talks about cells in roots that contain these special cells that have amyloplasts, organelles that are concentrated with starch, organelles that contain starch granules. These starch granules are pulled by gravity. And as a result, they activate sensory receptors, pressure-related receptors. These receptors, therefore, determine the way we are going to distribute oxygens throughout the plant. Very important. You need to understand how this hypothesis works. The hypothesis tells us that the distribution of the amyloplast is what activates these sensory receptors, and these receptors in turn determine how oxygen distribution is going to be achieved. That is what we see there, that the distribution of oxygen is going to depend on which side is down and which side is up, depending on the location of the amyloplasts and the receptors that have been, at the pressure receptors that have been activated. Okay. After understanding that, we can proceed to look at hydrotropism. Hydrotropism, if we go to the next slide. Hydrotropism. Hydrotropism, as you hear the word hydro, entails response to water. Yes, response to the direction of water. Plants need to respond to the direction of water. They need to grow towards water, because unlike human beings, plants are unable to walk to the water source. They therefore have to grow to the water source. So roots in plants are positively hydrotropic, implying that roots grow towards water. Roots are positively hydrotropic, implying that roots grow towards water. Implying that roots grow towards water. Yes, you can see in that image there, we have one with moist soil on my left hand, on my right hand, sorry, and one with dry soil on my left hand. When you look at the image of the right hand, we have the roots growing normally because the soil is uniformly saturated with water. However, when you place a plant, if you place a germinating seedling in a box with dry sand and you place a source of water in one region, you place a clay pot in one region, that plant is going, the roots of that plant are going to germinate towards the source of water. This indicates that roots are positively hydrotropic. Shoots, on the other hand, grow away from water. Shoots, on the other hand, grow away from water. You can't find leaves growing down to the water source. No, leaves grow away from water. This is negative hydrotropism. Yes, so under hydrotropism, we have response to water. We have response to the source of water. You either grow towards it or you grow away from it. And finally, I want us to look at chemotropism. Chemotropism is a growth response of plants to the direction So these are growth movements of plants. Growth movements of parts of a plant in response, yes, in response to a particular chemical. In response to a particular chemical. The common example we have here, in response to a particular chemical. The common example we have here is the growth of the pollen tube down the style during fertilization. Please put for me that slide. When you look at that slide right there, it's an extract from a flower. Yes, some parts have been removed, but what I want to focus on is the pollen grain on the top and the pollen tube. When you look at the pollen tube, we know from double fertilization that the pollen tube germinates from the stigma through the style towards the ovary. What drives this germination is chemotropism. The pollen tube germinates in response to a chemical. It grows towards a chemical as chemical that is synthesized by the ovule. In the absence of this chemical, the pollen tube will not be able to germinate towards it. The pollen grains do not germinate in the absence of this chemical. So, and a good example is the growth of pollen tubes down the style during fertilization in response to the presence of sugars in the style. The sugars in the style, the sugars in the style keep on causing the pollen tube to grow downwards until it reaches the ovule. What have we looked at today? Today we have talked about photoperiodism, we have talked about geotropism, we have talked about hydrotropism, and we have talked about chemotropism. We have talked about the different classes of photoperiodism. We have talked about short day plants, long day plants, and day neutral plants. We have said also that under these, the major factor we look at is the phytochrome pigment. The phytochrome pigment exists in two types. We have phytochrome red and phytochrome far red. Phytochrome far red is the active substance in this phytochrome system. The concentration of phytochrome far red determines different growth. Large concentrations inhibit growth inhibit flowering in short day plants, and short concentrations inhibit flowering, and large concentrations stimulate flowering in long day plants. We have talked about geotropism. We have talked about the Statolis hypothesis. The amyloplasts causing growth in particular directions. We have talked about hydrotropism, and we have said roots are positively hydrotropic. They grow towards water. And we have concluded with chemotropism, the response of plants to chemicals. I have been your teacher, Mr. Lwanyaga Solomon. Thank you for being good viewers. Have a great day.
Television, your family network. Omusumba Jackson Senyonga ategeeza abantu mwenna nti kati musobola okugoberera service za Christian Life Church okusaba copy 5 wamu ne programu za Pastor Jackson Senyonga ez'enjawulo ez'oku TV ne radio ng'oyita ku mikutu gye mugatta abantu egy'enjawulo giyite social media platforms ku simu yo, kompyuta yo oba ekirala kyonna. Ku Facebook teekamu Pastor Jackson Senyonga Ministries, ate ku YouTube teekamu Pastor Jackson Senyonga Ministries, ku Airtel TV ate ku MTN TV genda ku Yo TV era oyinza okukyalira emikutu gyaffe nga www.topradiouganda.com ne Facebook page ya Top Radio 89.6. Kyalira emikutu egyo gyonna, wuliriza Top Radio osobole okusabirwa, okusumululwa, okuwonyezebwa. Kino nno kikoleddwa olw'okwagala okutuusa ku buweereza wa Christian Life Church live woli mu kaseera kano ng'eggwanga lyonna ligezaako okwetereeza olw'okutangira akawuka ka coronavirus obuteegazaanya mu ggwanga Uganda.
School ly'essomero erya secondary erya O ne A-level eryazimbibwa ku musingi ogw'okutya Katonda nga tusomesa day ne boarding era nga tulina ebisulo eby'omulembe. Tulina abasomesa abakugu abeetegefu okusomesa abaana mu misomo gyonna egya arts ne sciences. Tulina computer lab, science lab, ekisaawe eky'omulembe okukosa ne clubs ez'enjawulo omuli omupiira, okudduka, okubaka ne MDD. Okusomesa kwaffe kuli ku mutindo gwa nsi yonna. Tulina clinic era nga mulimu omusawo omukugu omutendeko obulungi. Tusangibwa Seta Bajjo kilomita 1.5 nga wakava ku Jinja Road mu trading center y'Ekigunga. Okuwandiisa abaana kugenda mu maaso, tukubire ku simu zino